Here's a genus of orchids called Paphiopedilum. It has guard cells. It has stomata. The guard cells have I think they have chloroplasts, or the vestiges of chloroplasts, but they don't have chlorophyll. The guard cells lack chlorophyll in this group of plants. These plants don't respond to red light. They still open in response to blue light, but they don't have a response to red light at all. That's pretty good evidence that chlorophyll is involved in that red light response, if you ask me. And somehow these plants get along just fine without the rain. Of course, they won't grow on the prairie in Nebraska, but they grow someplace. All right, that's pretty interesting. That's pretty interesting. On the other hand, what about going the other way? Here's a fern called Adiantum. You know this one? Northern maidenhair fern. Anyway, this is a genus of ferns. Stomata of Adiantum don't respond to blue light, but they do respond to red light. They don't have a blue light response, a typical blue light response, but they have a red light response. It's backwards of the other one. They get along just fine too. The red light response is inhibited by DCMU, just like we've been saying. And these guard cells are chock full of chloroplasts. They have more than usual chloroplasts, more chloroplasts than usual guard cells. And they have some other weird things that happen in there. Okay?
So, can I claim that the orchids is resistant to DCMU? Can I say that?
Well, you could say that the guard cells of these orchids don't respond to DCMU. DCMU has no effect on the guard cells and stomatal opening in response to blue light. Yes, you can say that. However, DCMU will still inhibit photosynthesis in the other cells of the plant and still kill the plant. But the guard cell itself won't be affected by it. Just all the other photosynthetic cells will be affected by it. Yeah, you can say it. Right. And you could say also that guard cells in this plant aren't affected by dithiothriatol, which I get to in a minute. Okay, but again, the fact that these plants exist and live, and the fact that these plants exist and live and do just fine, again, is evidence that they're two separate systems that we're talking about for the light responses to mono. Okay. There's red light for you. Now, what about blue light? There's not as much known about blue light response, although more and more information is becoming available. And in fact, Eduardo Zeiger, one of the authors of this book, that's been his whole research career, is stomata, and in particular blue light response of stomata. So there's a fair amount of information in here, at least up to the time when this thing was published, which is 3 years ago. Okay, blue light is a total separate response. The photoreceptor for blue light response is a carotenoid called zeaxanthin. Zeaxanthin is found in photosynthetic cells, and in fact is part of what's called the xanthophyll cycle, which is the interconversion of zeaxanthin to violaxanthin and back again, depending on environmental conditions. Those pigments interconvert and have different absorption spectra, and the expert on that is sometimes here, John Gammon in the School of Natural Resources. He's done lots of research on xanthophyll cycle and remote sensing of the state of the xanthophyll cycle. Anyway, blue light stimulates proton extrusion from guard cells. And it stimulates them via a plasma membrane bound ATPase. So there's in the plasma lemma, in the plasma membrane around the guard cell are these ATPases. And when the guard cell senses the blue light, when zeaxanthin knows that the blue light's turned on, those ATPases break down ATP into ADP and use that energy to pump a proton. So that starts the whole process of motion for small. In some plants, green light inhibits the blue light response. Don't worry about it, let's not overly complicate things. This is complicated. Okay, so there is experimental evidence that implicates zeaxanthin in the blue light response of stomata. By the way, you know, when I've taught this course for many years, maybe longer than some of you have been alive. I don't want to get into how old you are, but I'm old enough to have taught it for 29 years. When I first started teaching this course, none of this was known. Blue light was just, yeah, there's a blue light response, but we don't know anything about it. Now, these gaps are being filled in, right? Anyway, zeaxanthin is the photoreceptor for blue light response. What evidence do we have? Well, again, we can look at the absorption spectrum of this pigment, zeaxanthin, and the action spectrum for blue light stimulated stomatal opening. Here we go. Here's the action spectrum for stomatal opening here without any numbers, but here's the wavelengths, 350, so 400 is about what your eye will see, 400 to 500 nanometers. Blue, getting into a little bit of the greenish yellow part of the spectrum. In this, and if you look at the action spectrum for stomatal opening and compare it to the absorption spectrum for zeaxanthin, it matches somewhat. I mean, it's not a perfect match, but you know, this is science. It's not exact. You know, science isn't an exact thing. People that aren't in science have a misperception, I think, in general. It's stereotyping everybody in the world, right? And have a misperception of science as being so exact and know everything and take all the fun out of stuff, right? But that's not how science works in my experience, anyway. Yeah, there's a lot of knowledge that's known, but every time you learn something, it raises 2, 3, 4 other questions and keeps you going, right? It's called job security. Think of the sciences. Anyway, you know, it kind of matches. It's not too bad of a fit. Let's put it that way, right? Okay, so that's some circumstantial evidence that zeaxanthin. If you look at the daily course of stomatal opening in intact leaves, you can measure stomatal conductance over the course of a day, and I showed a slide of that earlier, looking at potassium and sucrose concentrations in guard cells and things. Remember that? I should put that on here. The daily course of stomatal opening in intact leaves. If you plot the stomatal conductance, it matches well with the zeaxanthin content in the guard cells. So when they're open, more zeaxanthin. When they're closed, less zeaxanthin. Again, circumstantial evidence, but all these things taken together starts to make a case for implicating zeaxanthin as the photoreceptor. Blue light stimulated stomatal opening is inhibited by this chemical dithiourietol, DTT. I don't know what it looks like, something. DTT, okay. So in the xanthophyll cycle, we have viola, xanthin, anthero, xanthin, zeaxanthin. These are pigments found in the chloroplasts. And they're carotenoid pigments, right? They're all different kinds of carotenoids. They're all different xanthophylls, actually. And normally, these things interconvert depending on the environmental conditions. So in high light, I'm pretty sure how it works is in low light, it's mostly viola xanthan. And in high light, you get a lot of zeaxanthin. This gets converted to zeaxanthin. And this has different absorption characteristics than this, which also is an interesting phenomenon and has implications for how plants work. But anyway, this chemical, dithiophriatol, doesn't, it inhibits this interconversion. So the viola xanthan stays like it is, and the zeaxanthin doesn't increase. That chemical inhibits the blue light response of stomata. Well, that's another bit of evidence, right? How about this one? Here's another one. Last one I got. Mesembryanthemum crystallinum. You know this plant, the ice plant? That's from, I mentioned it too with the pressure probe, remember? This was one of the first plants that they poked that pressure probe into because it has, some of the cells in it are really big. But anyway, you know, it's native to South Africa. Used a lot. I used to live in San Diego. It's used a lot on the sides of the highways there to hold soil down, just like the native C4 grasses are used here in Nebraska. But in San Diego, ice plants used because it's quite drought tolerant. Won't grow in Nebraska over the winter because it can't handle the cold. But anyway, this is a facultative CAM plant. Remember, we have C3 plants, C4 plants, and CAM plants. They're crassulacean acid metabolism plant. It's facultative. That means if it has a lot of water, it does photosynthesis by C3 scheme, like most plants do, and just uses Rubisco and does it that way. But if things dry out, it'll switch to CAM mode. And remember how CAM plants work is they keep their stomata closed during the day. All day long, when the light's shining, their stomata are closed. And then at night, they open the stomata. And that's when they fix carbon, is at night. So they're very water use efficient, these plants. Not very productive, but very water use efficient. Anyway, bad conditions, this plant works like a CAM plant. And in good conditions, it works like a C3 plant. That's what facultative. Okay, so the guard cells accumulate zeaxanthin and show a blue light response when it's working in its C3 mode with lots of water around. But when it switches to CAM mode, the guard cells don't accumulate zeaxanthin and they don't show a blue light response. This is within the same plant. It can switch back and forth like this. So again, zeaxanthin, it appears to be play an important role in the response of guard cells to blue light. Okay, again, circumstantial evidence all the way around, but start to accumulate enough of it, and maybe it's true. So then you got to figure out all the messy little details of how this is important. Okay, so here's our action script. And this comes from an earlier version of this Plant Phys book. But you know what? If you look at the latest version, it's not much different. So if I can find it. It's not just white, it's got colors to it now. But if you look at this figure in the book, figure 10.13, it's essentially the same figure. but, you know, drawn a little flashier. Anyway, so in the chloroplast, here's the xanthophyll cycle, phytoxanthin, zeaxanthin. This is a mutant, you know, that lacks this, it's MPQ1. And that's been, mutants, behavior of mutants have also been used to study this blue light response, just like everything else. But somehow, blue light is sensed by this pigment here, zeaxanthin. That sets into motion something that moves out of the chloroplast to the plasma membrane. It has to do with this 14-3-3 protein, whatever that is, that binds with the ATPase and activates the ATPase at the plasma membrane that causes protons to be pumped out. Okay, so this is essentially the state of our knowledge of the blue light response, the mechanistic basis of the blue light response, right? So you can imagine if you're a biochemist that's interested in this, there's a fertile ground for exploration here, right? And people are working out the details of, you know, few years before this part wasn't even known. But you know, keep an eye on this topic because there's a lot of interesting stuff to discover. Lots of room to work if you want to get into biochemistry. We have a pretty good biochemistry department here, right? Down in the fetal center. Or you can stay here and work on whole plant stuff like most of us in this building are interested. Okay, but anyway, somehow something moves from in the chloroplast, set into motion by something happening with zeaxanthin, and that activates a ATPase on the plasma membrane that pumps protons out in the presence of blue. And that's really about what's going on. Notice here, CO2 sensing by Rubisco. All right? That's light. Blue light response, red light response. Different systems responding. The actions are the same. Protons get pumped out of the guard cell. Potassium and chloride concentrations increase in the guard cell. Malate concentration increases in the guard cell. The osmotic potential decreases. Total water potential is in disequilibrium with the total water potential around it. Water moves into the guard cell in response to that disequilibrium and total water potentials. The volume of the guard cell increases, its pressure potential increases, and the cells deform and open up a hole. Right? Any questions about light? Next time, instead of starting this and trying to go through it quickly, let's begin with this next time. Right? Any questions about it? Your exams? That's how people have a smile on their face, you know? What's the detail? Okay. Let's pass these out, and again, if you have questions about how I've graded or if I missed something or totally possible, let me know and we can discuss it. Otherwise, let me just tell you that the average was 91 and the standard deviation is 12. So again, have questions, please let me know. Luana? And I'm learning people. I Colin, he's not here, right?
Yep, he went to Baltimore, I think.
Baltimore. Oh, for ASA? This is ASA week. Amy. Zach.